Welcome to Lab 3. This video will help you prepare for all of the things that you'll be learning in the lab this week. This is going to include an introduction to histology. Recall that histology is the study of tissues. In this lab, you'll be learning to recognize 20 different tissues. We'll also be taking a quick look at basic structures in the skin and also introduce a microscopy technique and topic called the field of view. So here is a slide that should be familiar to you now. That is, we're looking at the hierarchy of how the human body is arranged. Starting off with the atomic level, we know about atoms. For example, sodium atom, carbon atom, oxygen atom. You've been hearing about this in lecture as well. You know, those atoms are what combine to make molecules. Molecules are then the basic building blocks of macromolecules, and you've been told about those 4 levels or those 4 different types of macromolecules in lecture. Of course, there are proteins, carbohydrates, lipids, and nucleic acids. And those are the 4 basic types of macromolecules. Those macromolecules are then the important building blocks of organelles, things like the endoplasmic reticulum, the Golgi apparatus, the plasma membrane, the nucleus, the components of the cell. You'll be hearing a lot more about those components in Chapter 3 of lecture. Now, those organelles are the building blocks of cells. There are over 200 different types of cells found in the human body. Those cells then combine to make tissues. Now, this is really where we are today in our lab. You're going to be looking at cells under the microscope that combine to make tissues. There are 4 different types of tissues. Those 4 different types of tissues include epithelial, connective, muscular, and nervous tissues. And then those 4 types of tissues combine to make organs. If I were to hand you a heart, a brain, a kidney, you would soon be able to recognize and identify many of these different types of tissues that compose that organ. And then finally, those organs combine to make the organ systems, or the body systems, of which there are 11. The respiratory system, the circulatory system, the endocrine system, the skeletal system, etc. And you know by now that those 11 systems are then the building blocks of you and me, the organism. So you've seen this hierarchy from atom to atom in lecture, and here now is a different presentation of that same information. So I mentioned there were 4 types of tissues. Let's take a little closer look at this information. So epithelial tissues. You're going to be learning and recognizing 7 different epithelial tissues. Epithelial tissues are composed of tightly arranged cells or tightly packed cells. They cover organs. They're going to be the major cells type and tissue type that form glands, like your salivary glands, your thyroid gland. And they serve to protect, secrete, and absorb. Where would you find epithelial tissues? You would find them covering organs, lining body cavities, as well as forming glands. The second type of tissue that you'll be looking at today are the connective tissues. And we have 9 different connective tissues for you to examine. We'll go over those specific differences between epithelial and connective tissues in a moment. But these are specialized tissues, but man, there's a lot of them. And they all have slightly different roles in your body. We'll introduce those. In your body, there are 3 different types of muscle tissue. There is skeletal muscle, cardiac muscle, and smooth muscle. Again, we'll go through those details. You'll get to recognize each of those. And then we're going to give you a quick look at one nervous tissue slide. So these 20 tissues will be your focus for this lab, learning to identify them, learning where in the body they're located, learning some special characteristics about each, as well as understanding how these tissues then become the building blocks of organs. So histology, for many students, is a brand new topic of study. You may have in high school or other science classes taken a look under the microscope and looked at cells. However, histology is for most students a whole new world. Part of that reasoning is, one, you've not used a microscope much before. But perhaps even more of a struggle is appreciating that when you are looking at histological samples, you're looking at a slice through an organ oftentimes. You're looking at a slice through the heart, or a slice of tissue through skin, or a slice through the liver. And so how the cells are arranged in that random slice can have a significant impact on what one sees when looking through the microscope. So just taking a look at this short bone, this little bone that you see here, you can see that depending upon the way in which that tissue is sliced, it can have a significant impact on how you see it. So a bone sliced longitudinally, as you see here, would give what would probably be recognized as a bone. If you cut it in a cross section, right? Cut it in a transverse section, you would see a round object that again doesn't seem odd. And if you sliced it at an oblique angle, then you would see more of an ovoid section on the slide. Now, if what you're examining is a tube-like structure, like a blood vessel, or a cavity of some sort, then a longitudinal view, let's think about this, would only show you this. You would really not have much perspective of that tube. Whereas a cross-section would give you a nice circle, and again, an oblique would give you an ovoid-shaped image. So again, how the tissue is sliced makes a big difference in how one sees the tissue. So getting used to this will be important. Now let me just say straight up that we're not here to trick you on this. The images that we will share with you and expect you to know are, quote, standard, ideal images. These are images that are going to come from a number of sources: your textbook, your lab atlas, Mastering A&P, the lab manual, or PAL, the Practice Anatomy Lab program that's a part of Mastering. So as you look at these different sources and review these different tissues, the images that we will have you identify are ones that look identical to those or are extremely similar to those tissues. It will be important that you are able to differentiate them, but we're not going to be giving you odd angles or obscure tissues to examine or to identify. So let's start off with epithelial tissues. We're going to break this presentation up in order: epithelial tissues, followed by connective tissues, followed by muscle tissue, and then finally nervous tissue. So in dealing with epithelial tissues, there are two important features to be looking at. One is the shape of the cells, and number two is the number of layers of cells in that tissue. There are three basic cell shapes. Epithelial cells can be squamous. You'll hear squamous as well. I think squishy, squashy, flat cells. Cells can be cuboidal, right? That is, they form cubes. Now, in 2D, it's going to look more square-like. And cells can be taller than they are wide. That is, they form rectangular prisms or look like a rectangle. Secondly, cells can have different layering patterns. And so an epithelium that has only one layer of cells is said to be simple. A tissue that has two or more layers is said to be stratified. And we'll deal with this pseudostratified in a moment. One thing I want to introduce to you right now is this dark line at the bottom of the epithelial cells. So that brownish line, I've now made it black, is called the basement membrane. And the basement membrane is a layer upon which the epithelial cells sit. And below the epithelial cells and below the basement membrane will be found a layer of connective tissue. Okay, so here we see on the stratified tissue, there is the basement membrane. Here are many cells that make up the epithelial layer. Because there's multiple layers, we're going to call that stratified. And below that basement membrane, deep to that basement membrane, one will find connective tissue. Now, the pseudostratified columnar tissue, which we'll be looking at today, here's that basement membrane. Here are the epithelial cells above. Here's what's odd about this tissue, is that it's called pseudostratified, and that name should tell you that it is falsely stratified. At first glance, it looks like there's a couple of different layers of cells, right? It looks like there's a layer of cells here and another layer of cells higher up. But in fact, if you look at this more carefully, if you could go in and take a look, every cell here is actually connected down to the basement membrane. So while it looks like there's more than one layer of cells, every cell is adhered individually to the basement membrane. So it is called pseudostratified. And because the majority of those cells are tall cells, this phrase goes together, pseudostratified columnar epithelium. Now, there are 5 important characteristics of epithelial cells that I want you to know. One I've already mentioned. They are tightly packed. Okay, starting at the bottom here. So these cells are tightly packed. They are touching each other. That means that there's very little extracellular matrix. Extracellular matrix. Very little ECM. Very little stuff outside of the cells because the cells are all touching each other and are tightly packed together. Number 2, I've already shown this to you as well. These cells are found on top of, superior to, connective tissues. Remember, the epithelial cells sit on the basement membrane. Below that, we find connective tissue. Number 3, these cells are constantly dividing. They are highly mitotic. And what that means is that they're undergoing mitosis very quickly. If they are damaged, they are very quick to repair themselves. You know, if you burn your tongue, that within a day or so, it seems to be fine. If you scratch your skin, that your skin regenerates very quickly. And that is true of perhaps if you were to bite the inside of your cheek, you know that those epithelial cells inside your cheek are going to replace and repair quickly. Number 2, they lack blood vessels. Epithelial tissues lack blood vessels. This makes sense. If you scratch yourself, you don't bleed, right? When you defecate, you don't bleed. When you eat Doritos, right, and those sharp edges perhaps would scrape down your esophagus as you're swallowing, you don't bleed. So it's a really good idea, right, that we don't have blood vessels sitting in our epithelial layers. And then finally, there's always a free surface. That free surface, you will see the word apical. But these tissues have an apical surface. That is, they have a surface that's open either to the inside of a tube, or they have a layer that's open to the environment, right? If you're thinking about your skin, the outer layer of your skin is open to a free surface. So keep those characteristics in mind. You'll be comparing and contrasting those tissues with other tissue types as we move forward. So there are 7 epithelial tissues you need to know. There are actually a few more than this list includes, and those you can ignore, and we'll have you skip over those in lecture and in lab. We're going to break these up into groups. So we have 4 different simple epithelia and 3 different stratified epithelia. So without even looking at these more specifically, if I said a simple squamous epithelium, what are you expecting to see? Right? There's going to be one layer of cells, and those cells are going to be flat, right? Squamous. What about simple cuboidal? Correct. One layer of cells that are cuboid in shape or square in their two-dimensional appearance. Simple columnar. One layer of tall, column-like cells. And then that pseudostratified columnar, we've already talked about that. That one is, while it appears to have multiple layers, is a single layer of cells, most of which are tall. On the stratified side, there is stratified squamous keratinized, stratified squamous non-keratinized, and transitional. In all three of these, you're going to see multiple layers of cells, and I'll describe these to you as we get to them each in a moment. So let's take a look at these cells, at these tissues. So simple squamous. I've got two different views of this here. Let's start off with the one on the left. So simple squamous, I've got two arrows here for you. So basically I'm asking you to identify this layer of cells that's between the two arrows. And as you can see, there are cells in here, right? You can kind of see the nucleus. These cells are touching each other. They have, it's one layer of relatively thin, flat cells. So that is a simple squamous epithelium. Now imagine, however, though, that you were looking down on this tissue from the top. If you were looking down, a superior view, looking down or a top view on this tissue, now that would look more like a cobblestone, more like a brick pavement. And so what you have here are cells, and I'm not being perfect in my drawings here, but you've got a group of cells. They're all touching each other, and they are creating a layer. And that's what epithelial cells do. They create layers. And so looking down at this, you would see more like a cobblestone appearance. Looking at it from the side, you would see what's here on the left. Now, in addition to recognizing these tissues, it's also important that you know where they are found in the body. So one place that simple squamous epithelium are found are in blood vessels. So we can add that here to our list. They are found lining blood vessels. You'll also find simple squamous making up the air sacs of the lungs. The specific name for that are the alveoli. Alveoli, or each alveolus, those are the little air sacs in your lung. They too are surrounded by simple squamous epithelium. And then thirdly, mesothelium. Last week you learned about the three serous membranes, the pleura, the pericardium, and the peritoneum. Well, those three tissues are also referred to as mesothelia, okay, the mesothelium. So the mesothelium is just a code word for the three P's, right, the three serous membranes. So if you think about the visceral pleura or the parietal pleura, it is a layer of epithelial, simple squamous epithelium that lines the surface of the lung, or the layer that lines the surface of the thoracic cavity. So that's number one, simple squamous. Number two, simple cuboidal. Let me draw here the basement membrane. So I'm drawing on the basement membrane here. This is a circular arrangement of simple cuboidal. And you can see I'm drawing basement membrane in a number of places. And then you see cells, right? One layer of cells, and these cells are rather cuboid in their shape. Okay, and there's the nucleus. Now, this is the type of tissue found in the ducts and tubes of your kidney. There are also great examples of simple cuboidal in the thyroid gland and making up the liver. But the one that we will see most often will be from the kidney tubules of the kidney. So the kidney is filtering your blood and then is exchanging ions and other materials that are eventually going to create urine. And so we'll see that this simple cuboidal epithelium is really important for that movement of molecules into and out of what will become your urine. Simple columnar. Again, let me start off by drawing the basement membrane, and you'll see that there's a difference here in the coloring of the stain. But right here, okay, and then here is one layer of tall cells. See this okay now? Now that I'm showing it, it's easier to see, isn't it? And below it would be connective tissue. Here's another basement membrane, and here's another layer of epithelial cells on the other side. What you're looking at is a folded over piece of the small intestine. Food would be traveling through here. This would be the lumen of the intestine. And the small intestine is lined by this simple columnar epithelium. Now, one thing that is not visible, easily visible, but I want to describe to you, is if you look on this tissue, you see this shadow, sort of this shadow right here on the apical, on the surface of this tissue. That is evidence of little projections, little folds in the cell called microvilli. Little tiny finger-like projections that add to the surface area of this tissue. As food is traveling through the digestive system, the job of your body is to absorb those nutrients. Now, if those, in order to absorb those nutrients, there has to be an increase in the available surface area for that quick absorption. The timing of your digestive system is perfect so that food does not sit around in your gut too long. If it did, it would rot and spoil and you'd get all gassy. And if nutrients go too quickly and you have diarrhea, then you also don't get adequate nutrients. And so the timing of your digestive system to work perfectly and normal requires an increase in surface area to absorb all those nutrients. One more extra feature that is found here are these cells. Here's one here. You see it, something looks a little different here. This is called a goblet cell. There's another one right here. And those goblet cells are producing mucus. Okay, the mucus is important to keep your digestive system moist, just like you have moist surfaces on your many of your other epithelial surfaces. Here is that pseudostratified columnar. This is our fourth tissue. Again, let me do the same thing. Let me draw that basement membrane. I didn't say this already, but remember that this presentation, these PowerPoint slides are available to you in the lab folder. And so hopefully you're following along with those images and you can be drawing on these things yourself. So we see these are all those little white, looks like little marshmallows. Those are the goblet cells. They're producing mucus to keep this tissue nice and moist. It looks like a jumbled layer of cells in here, but in fact, if we could, and we can't here, but if we could, we would find out that every one of the cells is actually connected down and attached to the basement membrane. Below the basement membrane, we would find connective tissues. And one important feature found in this tissue, on the surface, on the apical surface, you can see these cilia. Cilia are apical extensions found on the tissue, and this tissue is what is found in your nasal passages and going down your trachea. And those cilia are there to catch particulates. So as you breathe in, and as the air goes down into your trachea, heading to your lungs, it's important that the tissues pull away pollen and particulates and allergens and dust particles. So that's what we have here in the pseudostratified epithelium. You have cilia and goblet cells. Now, if we look at this cartoon representation, you can see why these cells are called goblet cells. Look at what the artist has done, right? It's almost like a little wine glass that you can see here, right? And those goblet cells are the ones producing the mucus. So here's another goblet cell here. So it's kind of nice to see it in this medical illustration, or this illustration of these cells. Okay, so that is the pseudostratified. Now that represents the end of our 4 simple epithelium. Let's now move to our stratified epithelia. Number 1, stratified squamous keratinized. You'll also hear it called keratinized, stratified squamous. So that word keratinized can go in the front or the back of this term. This is what you find on the surface of your body, on your skin, from head to toe. And for the first time now, we're going to see a much less flat basement membrane. So here comes the basement membrane. And you can see it kind of moving around here, up and down, up and down, up and down. Okay. Below it is connective tissue. And above that basement membrane, we see lots and lots of layers of cells. Roughly halfway up, you see something dramatically happen. Lower down, you can see that these cells all have a dark staining nucleus. And right about here, you see something change. Right? We'll talk about the layers of skin later, and the layers of the epidermis later. But above that line, you notice that the nuclei are gone. The cells are now empty shells, right? They have lost their nucleus. They're no longer dividing. They've lost much of their organelles. And now these cells are just flaky biscuits waiting to slough off, right? So most of the dust in your house is caused by you, right? All these simple squamous cells leaving and going into the atmosphere. So when you look outside your window and you see that dust, that is mostly you and the people with whom you live. So that is stratified squamous keratinized. These cells make a protein called keratin. Keratin largely helps to keep your skin waterproof. It is a collagen-like protein. It is also the protein that makes up your hair and your nails. Now, the tissue on the other side of the screen is not stratified squamous keratinized. And I wanted to put this side by side so you can see this. This is non-keratinized. And what the difference I want you to see side by side is that here's the basement membrane. There's the connective tissue down below. Here are the epithelial layers. But notice that these cells have their nucleus, and that nucleus goes all the way to the surface. You do not see dead flaky cells. Okay, you still see cells that show evidence of a nucleus at the surface. Over here, these cells do not have any nuclei showing at the surface. So that is the distinction that I want you to see side by side. The one on the right is a non-keratinized stratified epithelium. So let's look at that again. Again, when we look at stratified squamous non-keratinized, you're looking at places like the inside of the oral cavity, inside of your mouth, your lining of the tongue, the lining of the vagina, the lining of the anus. This is where one finds stratified squamous non-keratinized. Looking at this, here's the basement membrane, all the layers of cells, flat, squamous at the surface with nuclei present. Again, basement membrane, multiple layers of cells, and you can still see the dark staining nuclei at the surface. The last epithelial tissue is transitional epithelium. This is a distensible kind of epithelium. That is, it stretches. And this is what you find in a few places, but primarily in the bladder. So again, connective tissue down below the basement membrane, multiple layers of cells. Here's the difference though. In transitional epithelium, what is referred to as umbrella cells, these wide sort of umbrella looking cells, and not all of them, but some of them have 2 nuclei in them. They are said to be binucleated. So you're looking for those domed shaped umbrella cells. You're looking at binucleated cells. And this would be an example of the bladder in a relaxed state. As the bladder fills with urine, the epithelial layer would thin out and it would transition, right, from looking thinner to thicker, thinner to thicker. And so that is why it's called the transitional epithelium. Okay, so those are the seven epithelial tissues that you'll need to recognize and learn. Today you'll be practicing looking at those through your lab manual, looking at PAL, Practice Anatomy Lab, and Mastering. You'll also be looking in your e-text of your textbook, as well as your Hebert Lab Atlas to see representatives of these. You can also do a Google search. Just type in simple squamous epithelium, and you will be overwhelmed with images that are beautifully labeled for your study as well. Here's a list of the 9 connective tissues you'll need to learn to identify today. Blood, adipose or fat. There are 3 different types of cartilage. Hyaline cartilage, the most common type. Fibrocartilage and elastic cartilage. We'll look at the 3 of those. Bone, specifically this is compact bone. A tissue called loose areolar connective tissue. Dense regular connective tissue, and finally dense irregular connective tissue. Now, we talked about the characteristics of epithelial tissues. Let me fill you in on the characteristics of connective tissues. So connective tissues are composed of loosely arranged cells. Okay, so these cells are not tightly packed. The cells are loosely arranged. And whereas in epithelial tissues, there is primarily one cell type, right? Epithelial cells, a few goblet cells here and there. Most connective tissues are composed of different types of cells. So we're going to see, for example, that bone has osteocytes, whereas cartilage has chondrocytes, etc. I'll introduce these cell types as we go through the different tissues. In addition to those loosely arranged cells, because there is a lot of space in between the cells, there is stuff in between the cells. That non-living stuff between the cells is referred to as the ground substance. Now, the ground substance is really composed of proteins, primarily proteins. And there are 3 different types of proteins that you're going to hear described primarily in connective tissues. You're going to hear about collagen. You're going to hear about elastin. And you'll also hear about reticular fibers. Now, when you hear the word collagen, I want you thinking strength. Collagen is what gives connective tissues their incredible strength. Tissues like ligaments and tendons and bones are very strong because of their collagen content. Also, though, many connective tissues have a fair amount of elastin. Now, elastin is what gives tissues their stretch, gives their elasticity. So you can wiggle your nose, you can wiggle your ear. It's very bendable, right? Very pliable because of the elastin that is found in some of your connective tissues. And then reticular fibers. Reticular means network. And so these are fibers, these are proteins like collagen, but are really there not so much for strength, but more of like a scaffolding. More of like a scaffolding, okay? So when I talk about collagen, elastin, and reticular fibers, I think strength, stretch, and scaffolding. Right? Three S's to describe the function of these 3 different types of proteins found in the ground substance. Now, another term for the ground substance is the extracellular matrix. That is the stuff that is extra, outside of the cells, the matrix, the proteins outside of the cells. And then the functions of connective tissue, I'm not going to list them here, but there's a lot of different functions. Think about bone, right? Bone is very supportive, it gives structure, whereas blood, also a connective tissue, is providing immune function as well as carrying oxygen. And if you think about tissues like tendons and ligaments, they're providing a lot of strength and connection between different structures within the body. So the functions of the connective tissues is very broad. So let's go through these. And again, you're going to be seeing different views of these tissues between your lab manual, PAL, your lab atlas, etc. So here is a very close-up view of blood. You will not see blood any closer up than this view of blood. And what you have here, let's talk about the cell types, right? These are red blood cells. All these guys are red blood cells. RBCs, red blood cells. And this larger cell in the center is a white blood cell. And what I want you to see is that there's a lot of space in between the cells, right? Lots of space between the cells. So that is that ground substance. Now, blood is unique in that there's a liquid ground substance, right? The space in between the cells is actually a liquid called plasma. Plasma. And in that plasma, there are proteins. And so because of that, blood is a connective tissue. Loosely arranged cells surrounded by stuff called plasma in which there are proteins. And those proteins are involved in blood clotting and things like that. Now, one of the jobs of blood is to search out and fight off bacteria, but also, of course, we've got immune function, fighting off viruses. We also have carrying of oxygen and carbon dioxide. There are lots and lots of functions of blood. We'll be dealing with blood a lot more in the 5th exam of the semester. And you'll get to learn a lot about blood also in labs 8 and 9. Adipose, or fat tissue. Now the cells here are called adipocytes. I think I just spelled that wrong. We'll try that again. Okay, so the cells are adipocytes. Fat is a protective layer. It also helps to store some energy. What you see are cells that are largely empty looking. Now, in your body, they're not empty. But in the process of preparing these samples for view for the microscope, there is a step that involves that actually causes all the fat inside the cells to dissolve away. And so what you see are empty looking cells. The nucleus gets shoved up against the edge. You can see these dark staining nuclei here and there. And the cell is just an empty shell of what it normally would be. When you see these connective tissues, I think you'll agree they all look quite different from each other. And your job will be to recognize them, know the cell type, the adipocytes in this case. You may hear adipocytes pronounced as well. And also to have an understanding of where these tissues are found. Next tissue is one of 3 cartilaginous tissues. There are 3 types of cartilage: hyaline, fibrocartilage, and elastic cartilage. They all have cells called chondrocytes. Chondrocytes, the type of cells found in cartilage. Remember, chondro means rib or cartilage. And these cells are said to live in little spaces called lacunae. So here is a cell. Here's a cell. Here's a cell. You can see the nucleus. Notice lots of space in between, right? Very loosely arranged cells. And hyaline cartilage, hyaline means glassy. And so this is a very translucent type of tissue. It sort of looks like fine porcelain. This is the tissue that makes up the articulating surfaces, the ends of your bones, where two bones come together, where the joint is formed. If you've ever had a chicken wing, you all have, right? That little white ball at the end of the bone, that is hyaline cartilage. It's very clear. If you could scrape that off and look up through light, you would see that it's very translucent. This is the type of cartilage that makes up your fetal skeleton. This is also the kind of cartilage that you find, for example, making up your rib cage. Okay. When I see hyaline cartilage, or when I see cartilage in general, I see little eyeballs, right? So it looks to me like little eyeballs floating around here. The second type of cartilage is fibrocartilage. And this cartilage is not as ubiquitous. It's not found everywhere in your body like hyaline is. This cartilage still has chondrocytes, right? Those chondrocytes are still found in those spaces called lacunae. But the difference is that these chondrocytes produce a different mix of proteins. And the proteins that they make, primarily collagen. This collagen provides a tremendous strength to this tissue. Fibrocartilage is what you find in the cartilage of your knee, called the meniscus. It's also what you find in between your vertebrae, called the intervertebral discs. So this is a very weight-bearing, tougher sort of cartilage, right? Fibrocartilage. Thirdly is elastic cartilage. As the name suggests, this cartilage is more elastic. It is more flexible. This is the kind of cartilage you find in making up your ear, making up your nose, making up the epiglottis, a flappy structure found in your throat. Again, those chondrocytes, those cells called chondrocytes or chondroblasts, are actually making more elastin than other proteins. But you still see cells in that space, in that lacunae, still looking like little eyeballs looking at you. Bone tissue is one of the tissues we'll be learning the most about this semester because there's some really cool things about it. And there are 6 different things that I want you to learn about bone tissue. So let's take a look at this. When you look at compact bone, that is the bone, the outer layer of your bone, what you'll see is a circular arrangement. Okay, you can see a number of these circular arrangements. It kind of looks like a tree trunk. And there are many layers here that you can kind of see as you look at this. Many concentric layers. So each of those circular structures, those circular arrangements, is called an osteon. Okay, an osteon. These are actually cylinders that run the length of the bone. In the very center of that osteon is an open canal called and I'll draw it up here in the cartoon as well. That is the central canal. You may see it also called the Haversian canal. There are blood vessels and nerves that run through those canals. I mentioned those layers, so you can see that the tissue is made up of these layers. Each layer, each concentric ring is called a lamella, or many lamellae, A-E in the end. The bone cells are osteocytes. So cartilage has chondrocytes, fat has adipocytes, and the bone osteocytes also reside in these spaces. These spaces are called the lacunae. Okay, lacunae. So that's similar to cartilage. And there's a difference here, though. Bone, as you know, is a hard tissue, very tough. And the cells living in bone are still needing oxygen and nutrients and hormone signals. And so there has to be a way for these cells to communicate between this hard, bony tissue. And there are little canals that branch between the lacunae. Okay, so the first 5 letters tell you what it is. It's a little canal, canaliculi. Each one will be called a canaliculus, US. But many caniculi connect these cells one to another. So if we look, if I blow this up, you can see these what looks like stress marks, right, that travel in between these lacunae. And those are even better shown up here in this cartoon, but they're labeled the canaliculi. But these little lines are little canals, and that allows the cells to talk to each other. We'll deal with Volkmann canals in a future lecture, so don't worry about Volkmann canals today. Loose connective tissue, you'll also see it called areolar connective tissue, or altogether loose areolar connective tissue. This is a type of tissue. It is found pretty much everywhere throughout your body. It's very much a building block sort of tissue. The main cells here are called fibroblasts. And as this name suggests, these are cells that are making fibers. They're making proteins. Now again, the major proteins of connective tissues are collagen, reticular, and elastin proteins. So what you see here, here's the nucleus of a fibroblast, fibroblast, and all these stringy-like components in the extracellular matrix are collagen, elastin, and reticular fibers. I'm not asking you to know which protein is which, but to me this looks like silly string. It looks like confetti or some sort of crazy arrangement of pasta, right? And so that's what I look for when I try to identify loose areolar connective tissue. Dense regular connective tissue. Again, same idea. There are fibroblasts. They are making fibers, but the fiber that these cells are primarily making are collagen. And this collagen gets laid down in very regular patterns, To me, this looks like wavy bacon, right? It just kind of looks like a bacon. And these long waves that you're seeing are collagen fibers. And then the dark staining areas are the nuclei that are squished, okay? So you see dark staining nuclei of the fibroblasts creating long collagen fibers. Remember I said collagen is what gives tissues incredible strength. And this is what your tendons and ligaments look like under the microscope. Now this tissue is almost avascular, meaning it has little to no blood flow. And that's why tendons and ligaments are so difficult to repair. There isn't a lot of restorative blood flow going to tendons and ligaments. Going back, I forgot to mention one of the characteristics of cartilage. Cartilage is avascular. So when you look at hyaline cartilage or fibrocartilage or elastic cartilage, it's very translucent. Okay, it's very clear. There is no blood flow. And this is why if one has damage to their intervertebral discs or to their meniscus in their knee, it doesn't repair itself, right? Once that cartilage is worn down in an arthritic change, it does not restore itself. It's also the reason why if a person has their ear or their nose pierced, that there's relatively low blood flow, right? Because the cartilage of the ear has very little blood flow going through it. Our last connective tissue, dense irregular connective tissue. So here, let's talk about the comparison of these. This is loose connective tissue, right? The fibers are loosely arranged. They're all over the place. Whereas in dense regular connective tissue, the fibers are densely packed in a regular arrangement, right? We see those collagen fibers. In dense irregular connective tissue, there is dense packing of the fibers, the collagen, but in irregular patterns. So I see this as swirly whirlies. That's when I see dense irregular connective tissue, I kind of see swirly whirly kind of patterns. So what you're seeing here is that fibroblasts have made collagen, but in more random or irregular patterns. And this is what you find in your dermis. So in your skin, the outer layer is the epidermis. We've talked about that. That's where you have the stratified squamous keratinized layer. And then below the basement membrane, you have the dermis. And that is composed of this tissue type, dense, irregular connective tissue. So there are 7 epithelial tissues and 9 connective tissues for you to learn. Know where they're at in the body, know the characteristics, the cell types that I've described. In addition, there are 3 muscle tissues for you to learn: skeletal muscle, cardiac muscle, and smooth muscle. I'm introducing them to you here now. You'll see them again when we get to exam 3, when we discuss the muscular system. You'll see cardiac muscle again when we get to the heart in exam 5. But for now, you're going to learn to recognize these 3 muscle tissues, know where they're located in the body, know if they are voluntary or involuntary in their modality. That is, how do they work? Do you have actual control over them? That is voluntary, or are they involuntary, meaning that these tissues work around the clock 24/7 without your involvement, without your conscious control. So let's start off here with skeletal muscle tissue. This is a good representation of this. Now, when I see skeletal muscle tissue, again, this is what is making up the named muscles of your body. So as we get into a lab or two from now, and we're learning about the biceps femoris or the rectus abdominis or the frontalis muscles, any of the muscles that you'll be learning in the body, this is what they look like under the microscope. You see a very strong striping pattern. This striping pattern is what's called striations. So there's striated tissue, right? Striated tissue. And another thing to appreciate about this tissue is that what you are looking at in this view, you're looking at a portion of just a few cells. So these are long, skinny cells. Each cell is called a muscle fiber. Each cell has many nuclei. So you can see here there's 4, 5, or more nuclei. And we see the striations. So I just want you to think, when you see those strong striping patterns, think skeletal muscle tissue. The second type of muscle tissue is cardiac muscle tissue. Now, these cells are a little bit shorter, right? The heart is not as long as some of your long skeletal muscles. And the most important characteristic of cardiac muscle that I want you to get your eye to look for are the presence of these intercalated discs. Intercalated discs are a unique tissue to cardiac muscle, and they are these lines that you're seeing in this particular image. Okay. The cells are traveling like this. You see long, striated cells. But you also see these intercalated discs. Now, to me, this looks like bamboo. I mean, it looks like bamboo shoots, doesn't it? And these intercalated discs would be those, the lines that you see in bamboo. Now, the way I remember this is silly, but the way I remember this story is, what animal eats bamboo? Right? We see the bamboo appearance. What animal eats bamboo? We all think of panda bears, and of course, everyone loves pandas. So by love, you come back to the heart. Again, I just want to give you silly little, quirky little stories to help you learn how to recognize these tissues. The third and final type of muscle tissue is smooth muscle tissue. One of the characteristics of smooth muscle tissue is that there are no striations. There's no striping pattern here. Instead, it's smooth, right? There's a lack of those stripes that you see in cardiac or skeletal muscle. And the nuclei are typically these long, skinny, elongated, or cigar-shaped nuclei. So you see these nuclei of these cells. Looks like little boomerangs or little cigars. And to me, those long, squiggly-looking nuclei look like parasitic worms. And of course, if you had a parasitic worm, oftentimes it would be located in your intestines. And that's exactly where you find this muscle. This is the kind of muscle that helps with digestion, helps to push food through your digestive system 24/7. So we say that this muscle is involuntary. We have no control over it, just like your heart muscle. You have no control over your heart rate. And this is a smooth, non-striated muscle type. So think of those parasites. This is what's found again along your intestines, lining your stomach. And finally, the 20th tissue for this lab is nervous tissue. Now we are going to dive into nervous tissue significantly more detailed when we get to the nervous system in Lab 6 and in Lecture Exam 4. But for now, just know that the cells that make up nervous tissue are called the neurons. These cells are found all over the body, but there's the highest concentration of them in the brain and the spinal cord. And they are unique in that they're able to send electrical signals throughout the body very quickly to help with communication, reflexes, as well as maintain homeostasis. The second part of this lab is to learn just the very, very basics about skin. And so you are to learn just simply the layers. Don't worry about any other details. When we get to exam 2 in class, we'll deal more completely with skin structures. But for now, I just want you to know the 3 layers of skin. The outer layer is the epidermis. It is composed of, remember, this epidermis is composed of the stratified, squamous, keratinized epithelium. The middle layer, or actually the bottom layer, is the dermis. And the dermis, remember, was primarily composed of the dense, irregular connective tissue. And then that is all there is to your skin. Your skin is made up of the epidermis and the dermis. And then below your skin is a layer called the hypodermis. It's also called the subcutaneous layer. And that is primarily adipose, right? So this is primarily adipose. And that loose areolar connective tissue. So there's a good example. If I were to hand you a slice of skin, you will soon be able to say, okay, in the epidermis I see stratified squamous keratinized. In the dermis I see dense irregular connective tissue. In the hypodermis I can make out some adipose and some of that loose areolar connective tissue. In the lab we have a model that looks much like this. It's the same idea, but you do not need to worry yet about the other structures within the skin. Again, we're just going to be looking at those 3 layers, the epidermis, the dermis, and the hypodermis. Now, I keep saying the 3rd layer of skin. Every time you learn about skin, it will say epidermis, dermis, and hypodermis. But please, please, please know that the hypodermis is not a part of the skin. It's simply the layer on which skin sits. So in this picture, your skin would only go down essentially to that point, right? You would have the epidermis on top. You would have the dermis through here. That's your skin. And then all of this down here where all the yellow is shown would be the hypodermis. And that is not skin, but the layer upon which skin sits. Below that, right, you would see muscle. There would be muscle underneath that hypodermis. Okay, the last portion of this lab is an exercise using your microscope. It's called the field of view. You have a handout in your lab folder that goes over this with you. Read through that, and let me introduce this for you briefly, and then your instructor can help you with this as well. At the end of this worksheet, there are 2 pages of problems for you to work through, and the answers are all posted for you as well in the Blackboard shell. This is an important concept to appreciate. When you look into a microscope, the circle of light that you see is called the field of view. That's your viewing field, right? Your field of view. And when you are using the 4x objective, you are seeing things 40x times, right? You're seeing things magnified a total of 40 times. When you are using the 10x objective, you are seeing things 100 times fold in magnified. And when you're using the 40x objective, you're seeing things magnified 400 times. Now, we might call this the low or the scanning, right? This is the scanning or the low lens. And this would be the middle and this would be the high power. In our lab, we are not going to be going up to the 100x objective. That would be considered the oil objective, and we will not be using that this semester. So this field of view idea is simply this, and you're not going to get a chance necessarily to do this, but I want you to know that if you took a ruler and you put it down on the microscope and you looked under the microscope, when you are using the 4x objective, you are going to see essentially 5 millimeters across. We would say your field of view is 5 millimeters. So from edge to edge, you can see 5 millimeters of territory. When you flip over to the 10x objective, you now can only see 2 millimeters. And when you go up to the 40x objective, you can only see half a millimeter, okay? 0.5. So you only see half a millimeter when you are using the 40x objective. So that's an important concept to appreciate. Now, those are numbers you need to essentially memorize. That when you're using the 4x objective, you can see 5 millimeters across. When you're using the 10x objective, you can see 2 millimeters across. And when you can, using the 40x objective, you can see half a millimeter across. That's your field of view. Now, there's a mathematical relationship between these numbers. When you go from 4x to 10x, you are actually seeing things magnified 2.5 times, right? For example, if I said I'm going to go from 4x to 8x, right? That's twice as magnified. So if I go from 4x to 10x, I'm actually going to see things 2.5 times more magnified. Well, notice that there's a relationship here. 5 divided by 2.5 is 2, right? So when you go from the 4x to the 10x lens, you go from seeing 5 millimeters to 2 millimeters. And so you would just divide 5 by 2.5 to get 2. Likewise, when you go from the 10x to the 4x, from the 10x to the 40x total magnification, you are seeing things magnified times 4. So that means that the area you can see will be reduced or divided by that same factor of 4. So 2 divided by 4 is 1/2. So there's a relationship here mathematically between the lenses. Now, if this isn't making sense to you right now, it's okay. It will, I believe, as you work through the problems. So look in the lab folder. There's a field of view worksheet. There's another document that says field of view worksheet answers. And so work through those problems, do the best you can, check your understanding, and those answers are annotated. So it doesn't just give you the answer, but it actually tells you how the answer was derived. And I think if you work through that, you'll be fine. If after you've done that, it's still not making sense, please don't hesitate to ask your lab instructors for clarification. So here's why this field of view is so important and really an important skill for you to learn. This is the field of view, and it's telling you that you're using, this is the field of view 40x total magnification. So you know that 40x total magnification, you're using the 4x objective. We just said that when you're using the 4x objective from here to here, all the way across, remember, it's 5 millimeters. That's important to know. So the question is, how big is this object? How big is the cell that you're seeing in this view? Right? How big is this object? Well, we can make a guesstimate here, right? So how many of those cells would there be going across this, right? There'd be about 5 of those cells. Do you agree? There'd be about 5 of those cells if you put them all straight across. If this is 5 millimeters, that means that that one cell is approximately, right? This is just an approximate 1 millimeter. Okay? So I would always accept a range of acceptable answers for that. Okay, so work through those problems. And then finally, here's just a recap of what you need to learn from today's lab. Number 1, recognize the 7 different epithelial, the 9 different connective tissues, the 3 different muscle tissues, and the 1 nervous tissue that was given to you. Know where these are found in the body. Know the cell types found in each tissue and any special characteristics that were listed in this presentation. Finally, learn the 3 layers in the skin model. Again, really, I should say of the skin, really epidermis and dermis are the only 2 layers of the skin. The hypodermis is below that. And then finally, do that field of view worksheet. Lots of work for you to focus on this week, and I know your lab instructors will really help you make sense of all this. Have a great lab.